So, guys, gals, let's talk about the cell membrane. Question? What was that? What closed out? Sure, I can reopen the attendance. If I reopen the attendance, guys, gals, and you're not marked present for the second attendance, but you're marked present for the first attendance, it's okay. I want to avoid taking attendance twice in the same class period if possible. Minimize and keep open. So the attendance code is 3947. So let's talk cell membranes. As we're looking at the cell membranes and how to move things across the cell, we're going to talk about the plasma membrane. Our plasma membrane is selectively permeable. What does selectively permeable mean? It means it lets some things in and out, and other things, it acts as a gatekeeper. It doesn't let those other things in and out. Things that it'll actively let in and out, generally speaking, are going to be very small, non-polar molecules. A molecule of oxygen gas, a molecule of carbon dioxide gas. These things can just go straight through the phospholipid bilayer. Other things can be really small, but they have a hard time going across the membrane. A sodium ion, a potassium ion, really small, highly polarized particles have a hard time crossing this membrane. So today we're going to talk about how we get the easy things across, how we get the hard things across. Depending on what we're trying to move, we have a different technique in our body to move it across our plasma membrane. There are two big ways that we move things across. The easy way and the hard way. The easy way is called passive transport. Passive transport does not require ATP. I'll say that again because this is a key take-home idea. Passive transport does not require ATP. If you are thermodynamics buff, passive transport is powered by the law of entropy. It just happens spontaneously. Did I do that with my screen? Did I scribble on the screen? What is going on here? Let's try that again. Here's the clear eraser. Miss Londrich, are you logged in and are you pranking me right now? Okay. All right, I'm going to finish attendance. Oh, that's weird. Let's go ahead, go back to the presentation. Just ignore the random scribble on my screen. I have no idea how that got there. Because it wasn't there when I started the presentation, was it? And I was like, not at the computer. Okay, whatever. Moving on. Active transport requires ATP. During active transport, we're moving something against its concentration gradient. And because we're moving it against its concentration gradient, we're kind of moving uphill, so to speak, and it requires work. It requires the input of energy, or ATP. So let's talk about some of the easier forms of transport. Filtration is an example of passive transport. And we're just going to have physical pressure pushing something through the plasma membrane or through the membranes in between cells. So as we're looking at this diagram from your textbook, we have a blood capillary, and this blood capillary is leaky. There's little spaces in between the cells that make up the wall of the blood capillary. Those little spaces let little particles, little molecules get pushed through. Now with filtration, the more physical pressure we have, the faster something is filtered. I always like to think back to when I was a Boy Scout leader and I would take kids on camping trips. And I'd be, canoeing in a swamp and we'd have a little water pump and we'd be purifying water. And the harder you pushed on the pump, the faster the water would be filtered and the quicker you could get a drink. The same is true of filtration in the human body. Now, this pushing force in the human body is going to be blood pressure. So individuals, generally speaking, that have higher blood pressure will have a slightly higher rate of filtration. Individuals with a lower blood pressure will have slightly lower rates of filtration. Sometimes we'll just have simple diffusion. And when we look at simple diffusion in the bodies, this is just going to be something moving from an area of high concentration to an area of low concentration. This simple diffusion is an active process. We are going to find that simple diffusion happens spontaneously as the molecules are just bouncing around and spreading out apart from each other. My favorite example of simple diffusion is some kind of a spray perfume or spray deodorant. You spray it on your body. There's a super high concentration of the odor molecules around your body, and then they slowly spread out and fill the room, and then maybe they go down the hall. They will slowly diffuse to the areas of lower concentrations. When we look at simple diffusion, it does not require a filtration membrane or a selectively permeable membrane. So the simple diffusion happens within a cell. So a molecule, a substance can get through the plasma membrane, and then to go from the outer edge of the cell to the middle of the cell, we're going to have simple diffusion occur as this substance will move through the cytosol of the cytoplasm. So what can we do to increase the rate of simple diffusion? We can increase the temperature. This goes back to the kinetic theory of temperature. As molecules or particles are warmer, they move faster. So if we heat something up, we'll have a higher rate of diffusion. This is one of the principal methods for why we use hot water to cook our noodles as opposed to cold water, or you use hot water to make coffee quickly. And if you do a cold brew, you have to let it seep overnight because the rate of diffusion is a heck of a lot lower. Another thing that influences the rate of diffusion is molecular weight. Generally speaking, molecules with a smaller molecular weight will diffuse quicker. So things like oxygen gas, that has a relatively small molecular weight. A molecular weight of, 8 plus 8. So we're going to have a molecular weight of 32. Now on other things, bigger molecules like glucose, it's not quite a protein, it's not quite that big. Glucose will have kind of a medium-ish rate of diffusion. During your physio-X activities, you'll be working with really large protein molecules that have very large molecular weights. Those protein molecules with large molecular weights have a very low rate of diffusion. Something else that influences the rate of diffusion is how steep the concentration gradient is. If you have a sort of low concentration and a kind of low concentration, so they're almost equal to each other, there's not going to be that much diffusion between the two of them. But if you have a very high concentration and a very low concentration, there's a big gradient difference, and with that big difference in gradient, there'll be a faster rate of diffusion. That's one of the things you'll be working through in your physio-X activities. Membrane surface area influences the rate of diffusion. The higher the surface area, the higher the rate of diffusion. And then we also have membrane permeability. Some membranes are more leaky than other membranes. If it's more permeable, if it's more leaky, we have a faster rate of diffusion compared to something that's smaller or less leaky. This leads us to the diffusion of water. Water is such an important molecule in our bodies, guys, gals, that we have a special name for the diffusion of water, and that's osmosis. So when we look at osmosis, it's the movement of water across a selectively permeable membrane. And we are going to go from an area that has, a high concentration of water to an area with a lower concentration of water. So if we look at the figure, we start off with side A and side B. Side A has only water on it. Side B has water plus a dissolved solute. And over time, that dissolved solute causes water to be moved across the membrane. One of my favorite examples of osmosis is when I sprinkle sugar on strawberries. Sugar can be a solute. Strawberries have cell membranes. When I sprinkle sugar on strawberries, that causes water to be pulled across the membrane and be removed from the strawberry to the area where there's a lower concentration of water, where the sugar is. And then I get a nice candy glaze on my strawberries. Something that we need to emphasize with osmosis is that it draws or pulls water. You cannot push with osmosis. Osmosis will pull something across a membrane. So let's think of intravenous fluids and some applications. If we are going to give an intravenous fluid to a patient, we better make sure that intravenous fluid has the correct concentration of dissolved solutes in it. Otherwise, we're either going to draw water out of the patient's red blood cells, or we're going to draw water into the body cavities. A classic example of drawing water into a body cavity is when somebody is going in for a colonoscopy and they have to drink that big gallon jug of magnesium sulfate heptahydrate, and they drink it, and that magnesium sulfate draws water into their colon, and then they have amazingly explosive diarrhea for a night, and it cleans out their colon. That's an example of osmotic pressure. Another example of osmotic pressure would be maybe drinking seawater. For the record, if you're thirsty, don't drink seawater. Seawater has a higher salt concentration than your blood, and if you drink seawater, it pulls water out of your bloodstream into your intestinal tract and causes you to have loose stool, and actually will make you even thirstier than not drinking the seawater. Now, water in and of itself is a polar molecule. It has polar covalent bonds in it, and those polar covalent bonds of water make it really hard for water to move through the phospholipid bilayer. So we're going to have a special pore in the phospholipid bilayer, a special protein called an aquaporin, or a water hole. Aqua for water, pore for hole. An aquaporin is literally a hole for water in the cell membrane. And this aquaporin will speed up the rate of osmosis. So we can have osmotic pressure in the cell to move liquids around. That's a pulling force. We also can have hydrostatic pressure. Hydrostatic pressure is the pushing force. Osmotic pressure is concerned with salt or dissolved solute concentrations. The primary thing we're concerned about with hydrostatic pressure is going to be the fluid pressure physically pressing against the cavity wall. In human beings, the biggest source of hydrostatic pressure is ultimately our blood pressure. Occasionally, there'll be a homeostatic imbalance or some kind of a disease state where there's not enough of a balance between the hydrostatic pressure and the osmotic pressure. And we need balance. There's a give and take between the push and the pull of fluids in our bodies. And occasionally, we'll see individuals that have a lot of fluid buildup, usually in a leg, where they have edema of the leg, and the leg will swell up and get really big. A classic treatment for edema, in particular lymphedema, is going to be putting a compression bandage on the swollen appendage. Putting that compression stocking on the swollen appendage increases the pushing force, increases hydrostatic pressure, and causes more liquid to leave that appendage so that the swelling can go down. When we think of reverse osmosis, this was a term that confused me when I was a teenager and in my early 20s. You could think of reverse osmosis as filtration under pressure or hydrostatic pressure. Now, occasionally, though, you'll see this term used, reverse osmosis. This is just pushing something through a membrane to filter it. It's the opposite of osmosis. It's reverse osmosis. Osmosis pulls, hydrostatic pressure pushes. Now, in our bodies, we have different chemicals that can be dissolved. Some chemicals influence osmosis more than other chemicals. So as we're looking at these different chemicals, we need a way to quantify how much they influence osmosis. And there's a special unit that we use called osmolarity. Osmolarity is very closely related to molarity, which You covered in a chemistry class that you took as a prerequisite for this. When we look at osmolarity, the thing I really want to emphasize for you is that the number of ions dissolved influences osmolarity. The more ions you have dissolved, the greater your osmolarity is going to be, because the greater the influence on osmosis. So this time of year, we're all thinking about ice and snow, and we can think of different kinds of sidewalk salt. There's sodium chloride, and then there's calcium chloride. Sodium chloride and calcium chloride have different effects when we think of osmolarity purposes. Sodium chloride has a sodium and a chlorine ion. It's 2 ions for every unit. When we think of calcium chloride, we have 1 calcium and then 2 chlorides. So there's 3 ions for every unit. Different salts pack different punches. In particular, if you're looking at salts that have more than 2 ions present in them. So as we look at these different kinds of salts, these different salts have different effects in our bodies. Some things, like glucose, don't form individual ions when we dissolve it. Instead, there'll be that one glucose molecule. So glucose's impact on osmolarity is going to be a lot less. 1 molar glucose, or 1 mole of glucose, will dissolve into 1 mole of particles. 1 mole of sodium chloride will dissolve into 2 moles of particles, so on and so forth. As we're looking at osmolarity and the tonicity of the fluid that can influence the movement of liquids across the cell membrane. If we're working with a hypotonic solution, we're going to have a solution with a very, very low salt concentration, or a very low dissolved solute concentration. The classic example of a hypotonic solution is going to be deionized distilled water. And I remember one of the lab activities I did when I was an undergraduate was to take tadpoles, and we took frog tadpoles, and we put some in deionized distilled water, we put some in saline solution, and we put some in really salty water, let them sit for a while, and then recorded the mass of the tadpoles before and after. When we put those tadpoles into hypotonic solution, their cells sucked up water. They increased in volume. If we look at a red blood cell in a hypotonic solution, that red blood cell will swell up as a sphere. If we put something in a hypertonic solution, it's going to be in a very salty solution, or a solution that has a high dissolved solute concentration. Hypertonic solutions tend to pull liquid out of the cell, tend to pull water out of the cells. And that causes the cells to shrivel up. The technical name for a cell shriveling up, particularly a cell that does not have a cell wall, is going to be crenulate or crenate. And then finally, we have isotonic solutions. Iso meaning the same, tonic referring to the concentration of dissolved solutes. Isotonic solutions have the same concentration as the intracellular fluid, or the intracellular environment. So, from a clinical sense, if you're giving a patient a saline solution, you want to make sure that saline solution doesn't have a higher osmolarity than the inside of their cell. You don't want a lower osmolarity. You want exactly the same osmolarity as the inside of their cells. And if you have exactly the same osmolarity as the inside of the cell, then the cells won't swell up, they won't shrivel up, they'll just stay the same size. And for saline solutions, this is worth committing to memory because it's the most common solution. A 0.9% sodium chloride solution is the concentration that's isotonic with the human body. So, let's see if I can scroll down a little bit here. If we take a red blood cell and place it in a hypertonic solution, this means the concentrations of solutes in the solution is blank. Then the concentration of solutes in the intracellular fluid, and it will cause the cell to blank. So let's see how we're doing for comprehension. And let's go with 20 more seconds. Ten more seconds. So, if you put a red blood cell in a hypertonic solution, the liquid outside of that red blood cell has a higher salt concentration. It's going to have more solutes in it than the inside of the cell. And if there's more solutes outside of the cell, that's going to cause water to go from inside of the cell to outside of the cell. Water will leave the cell, and as water leaves the cell will shrink or crenulate as it becomes smaller and shrivels up. So as we're looking at the cell and the phospholipid bilayer that makes up the cell membrane, some particles need a little bit of help to get across. And those particles are going to have, generally speaking, some kind of a protein to help get them across. We're going to call this carrier-mediated transport, where we use a protein to get something across the phospholipid bilayer, the cell membrane. Now, these transporter proteins are designed to be either general or specific. Most transporter proteins, though, are going to be on the specific end of the spectrum. Now, as we look at these transporter proteins, they can only move something so quickly. There's a maximum rate that they can move a molecule or a solute across the cell membrane. This maximum rate is called transport maximum. And eventually, if you have too many molecules, too many particles that you're trying to move across the cell membrane, the transport maximum can't keep up to it. As we're looking at these transporter proteins are going to bind to the molecule and then transport that molecule unchanged, unaltered, to the other side of the membrane. My favorite example of transport maximum probably is going to be moving glucose out of the urine in the kidneys. And this is a really funny but kind of gross story. If you have glucose in your urine, that's not a good thing. For the record, it's bad to urinate out sugar. And if you have a lot of glucose in your urine, you're probably going to get a lot of UTIs as well. But if you have that glucose in your urine, your body is, sugar hungry. We're pre-programmed to crave and want sugar. So we're going to pull that sugar out of our urine and dump it back into the bloodstream. However, occasionally we'll have a patient with diabetes and they dump so much sugar into their urine that they can't pull it out quickly enough and they end up urinating out sugar. This condition of having sugary urine is called diabetes mellitus, and now we have glucose test strips and we can do it chemically, but 100 years ago, we did not have glucose test strips. And there was one way to test sugar concentration in the urine. They would taste it. Yeah. Think of that the next time you use a test strip and just appreciate how amazing the test strip is. So, as we're looking at ways to move something across the cell membrane, we have three different kinds of carriers, three different ways to transport something across the cell membrane. We have the uniport, symport, and antiport, which are just ways to categorize something based on how it transports the molecule. Uniport has that prefix uni in front. Uni means one. Uniport carriers move only one kind of solute. And then we have symport carriers. Symport carriers can carry more than one or two or more kinds of molecules at the same time in the same direction. Sometimes that's going to be called co-transport. A good example of this is when we build up an ion concentration in our bodies. And we use that ion concentration, either sodium or potassium or chloride ions. We can use those ions to carry another molecule against its concentration gradient. these symporters can harvest the chemical energy inherent of concentration gradients to move something else like glucose against its concentration gradient. And then we have antiport carriers. These are going to be protein carriers that move different things in different directions. The antiport carrier that has, by an order of magnitude, the most research done about it is the sodium-potassium pump in our body. The sodium-potassium pump you'll get very tired of talking about when we're done with the nervous system. When we look at the sodium-potassium pump, or talk about it a little bit right now, it brings sodium out of the cell and potassium into the cell. It pumps sodium out and brings potassium in. We'll talk more about that later. Now, these mechanisms can either be passive in that they're facilitated and they don't require ATP, or we can have active transport, primary or secondary active transport. And active transport does require ATP. It does require the input of energy. It moves something against the concentration gradient. So let's talk about facilitated diffusion. When we look at facilitated diffusion, it's going to be moving something with the concentration gradient. But this facilitated diffusion is typically going to be a particle that doesn't move against or doesn't go through the cell membrane easily. So we're going to need some kind of a protein to help transport it across the cell membrane. And that's where that transporter protein comes into play. We can also have primary active transport. When we look at primary active transport, that's going to move molecules across the cell membrane against the concentration gradient. Active transport moves against the concentration gradient and requires ATP. So these proteins that are moving chemicals against their concentration gradient are going to just very simply grab that molecule and push it across the membrane. It's primary. It will grab the molecule in question and push it across the membrane for the primary active transports. Two examples include calcium pumps or the sodium-potassium pumps. Those proteins will only move the molecules in question, or the ions in question. Let's talk some more about the sodium-potassium pump because it's such an important protein. The sodium-potassium pump in our body uses up half of your calories. One protein molecule moving ions in your bodies uses about half of the calories you consume on a daily basis. That's how important this protein is. Now, when we look at the sodium-potassium pump, it maintains very steep sodium ion concentrations and potassium ion concentrations. And these steep concentration gradients of sodium and potassium ions make it so that our membranes in our cells have electric charge to them. And different membranes have different kinds of electric charge. We use the sodium-potassium pump to keep our cells charged so that we have a membrane voltage built up across the membrane. Now, as we're burning through ATP and we're maintaining this electric charge across our cell membranes, we're going to have a lot of waste heat produced. And we don't waste all the waste heat. A lot of this heat is used to keep us warm. This is one of the reasons why we as mammals are endotherms. We maintain our own internal body temperature. And it's also a primary reason why we as endotherms, or mammals, need to eat so much food compared to reptiles or fish or organisms that don't maintain their own internal body temperature. We also have secondary active transport. These molecules these secondary active transport molecules aren't going to use APT directly, but instead use ATP indirectly. They'll use ATP indirectly by having that ion gradient. They'll build up a concentration gradient with the ATP, and then once they've used that ATP to build up the concentration gradient of sodium or potassium or whatever your favorite ion is, that concentration gradient of your favorite ion can then be used to move something else against its concentration gradient. The example from your text was the sodium glucose transporter. where we have a concentration gradient of sodium ions built up. In particular, that's built by the sodium-potassium pump. So we use a bunch of ATP with the sodium-potassium pump to build up a sodium ion gradient. Then that sodium ion gradient can be used to move glucose against glucose's concentration gradient. So that's why we call it secondary active transport. Instead of using the ATP directly, we will use it indirectly to make a concentration gradient, and then use the concentration gradient directly to move the molecule. So here we have a diagram from your text showing the sodium-potassium pump on the bottom margin, inferior margin of the cell, and that sodium-potassium pump is using the ATP, building up the ion concentration gradient, and then we have the sodium-glucose transporter on the other side of the cell that's using sodium ion concentrations to move glucose into the cell. So, let's test our comprehension. Which of these is an example of active transport? We have the diffusion of oxygen from a place of high concentration to a place of low concentration. We have facilitated diffusion of potassium. We have transport of glucose down its concentration gradient. We have the transportation of sodium from an area of low concentration to an area of high concentration, or we can move chloride ions following or with its concentration gradient. Which one of these would be active transport requiring ATP? We have about 20 seconds left. One of these is against the concentration gradient. All of the other ones go with the concentration gradient. So if you haven't answered yet, make sure you click answer. Again, we're only grading on participation. So it looks like as a class, this question was a little bit harder. The thing to emphasize with this question is that if you take sodium from an area of low concentration to an area of high concentration, for answer D, that's against the concentration gradient. And whenever you go against the gradient, that requires the input of cellular energy, or ATP. All the other options given were with the concentration gradient and did not require the input of ATP. Okay, let's talk about how to move bigger things across the cell membrane. We can use vesicular transport to take a large particle and move it across the plasma membrane. These are typical vesicular transport, usually will be used for things that are too big for a carrier protein to carry across the cell membrane. However, as we're using this vesicular transport, we're going to have smaller proteins in our cells that move the vesicle, a little storage sac, or I like to think of them as little suitcases filled with substances from one part to another part of the cell. And those little motor proteins are going to use ATP up in the process. One form of vesicular transport is exocytosis. Exo for out, cyto for cell. Exocytosis means moving something out of the cell. When we secrete a chemical from our cells, we are exocytizing that chemical from our cells. Then we have moving something into the cell. Endo for in, cyto for cell. Endocytosis is moving something into the cell. And there's multiple versions of endocytosis, multiple variations of endocytosis. So let's talk about those variations. We have phagocytosis. Phage, or phage, means to eat. And it's generally, when we think about it, When you eat something, it's typically going to be solid. So when we think of phagocytosis, we're moving large, solid particles into the cells. My favorite example of this comes from the immune system, which we'll talk about in Bio 314, where we have phagocytes, or white blood cells that specialize in swallowing bacteria and pulling the bacteria into the cell. There's a 1960s B-movie classic, The Blob. If you're looking for a good time, I recommend watching The Blob. It's about a giant phagocyte that consumes a town. We also have pinocytosis. Pinot, when I think of Pinot, I think of Pinot Grigio, a kind of wine. Pinot refers to, and I probably said it wrong because I'm from the Midwest and I'm not Italian. I know it's not pronounced Pinot Grinot though. So when we think of Pinot, think liquid, think drinking. Pinocytosis is to bring a liquid into the cell as a form of endocytosis. And then we have receptor-mediated endocytosis. And this receptor-mediated endocytosis is going to have very specific protein molecules on the surface of the cell membrane. And these specific protein molecules on the surface of the cell membrane are going to have their favorite particle. Whatever particle they're designed to attach to, their favorite particle will attach to these receptors. And when enough of those particles are attached, these particles will be brought into the cell. The receptor for receptor-mediated endocytosis is called clathrin. So that receptor on the surface of the cell is a clathrin protein molecule. Different kinds of clathrins are designed to attach to different kinds of particles. So here's a good example of a cell going through phagocytosis and exocytosis. We have a cell that finds a particle and it wants to consume that particle of foreign matter. It will extend a pseudopod to wrap around the cell and then wrap it in cell membrane and bring it inside. That phagosome, or that little suitcase that's wrapped up around the particle that was consumed, is going to bind to one of the organelles we're going to talk about on Monday, called the lysosome. And when that phagosome binds to the lysosome, that lysosome releases digestive enzymes. Those digestive enzymes chop it up into little pieces, and then those little pieces are processed, and eventually the waste products are exocytized and secreted as waste residue. As we look at receptor-mediated endocytosis, I want to include this slide so that you could see the clathrin receptors on the surface of the cell, and then as those clathrin receptors have their favorite particle bind to them, the cell membrane invaginates inward and then pinches off and brings your favorite particle into the cell. Whatever particle that specific kind of clathrin is designed to attach to will be brought into the cell. And then, last slide, I believe, transcytosis. Moving something from one side of the cell to the other side of the cell. So transcytosis is mixed up by filtration with students all the time. When we have transcytosis, instead of something going between cells, like we do with filtration, think of blood capillaries, and filtering liquids between the cells, with transcytosis, that substance is going straight through the cell. That's a key distinction. Filtration is in between cells. Transcytosis is through a cell. So with transcytosis, we will have endocytosis on one side, and then that phagosome will move the molecules across the cell cytoplasm and then exocytize them on the other side. Transcytosis inherently needs endocytosis paired with exocytosis. If you pair endo and exocytosis together within the same cell for the same chemical, That is transcytosis. I think this is a duplicate slide, but just for the sake of being clear, let's make sure we define exocytosis as secreting material out of the cell. And then one last review question and we're done for the day. So as we're looking at the cell, our cell membranes are permeable to blank, but impermeable to blank. What can we let through our cell membrane and what do we not let through our cell membrane? Small molecules can go through easily. Large molecules have a very hard time going through the cell membrane. So guys, gals, proteins do not move across the cell membrane. Proteins are large and have a hard time. See ya. Have a good day.